Okay, shall we start this fascinating subject again and carry on about HIV and AIDS infections? Now, this is the sort of things you're going to see in the clinic because nowadays we assay the patient's CD4 cells, we assay the level of virus in the bloodstream by PCR. So what are good predictors? Well, that set point that I mentioned, that's the level of virus production, the level of virus that you find in your patient's bloodstream after he or she has mounted that really quite strong immune defense against the virus is rather important. There's a good correlation between the number of HIV particles measured by PCR and progression to the disease. So if we look at viral load here, if you've got a pretty small viral load, then you're going to survive a longer time than if you've got a high viral load during this set point. So one of the things that's done with all patients is to look at their viral load. Now, CD4 cell numbers are not a very good predictor until later on in the course of the disease. So the viral load at the set point, measured by PCR, is a good predictor of how the patient is going to survive. One of the early manifestations of the AIDS epidemic was the increase in the incidence of a particularly virulent form of Kaposi's sarcoma. Now, not all cases of Kaposi's sarcoma, as I've already told you, are associated with an HIV infection. This was a disease that's been with us at low levels, particularly in immunocompromised people, for a long, long time. About 20% of homosexual HIV-positive males come down with Kaposi's, but very few of the other groups. So this doesn't look like it's the result of having HIV. And indeed, we now know it's because in the gay community particularly, another sexually transmitted disease, human herpes virus 8, otherwise known as Kaposi's sarcoma, associated herpes virus, is the actual causative agent of this problem. HIV is a cofactor. You've got immune suppression as a result of the HIV. This allows other viruses which were normally kept down by the immune system to now reveal themselves in the case of HHVH as a result of results in Kaposi's. EBV results in various lymphomas that come up. I mentioned multicentric Castleman's disease yesterday, caused by EBV. Actually, that's caused by herpes virus 8, isn't it? But there are other diseases which are associated with EBV that come up in the AIDS-infected patient. In the eye, cytomegalovirus causes retinitis, a major problem in HIV-infected patients, leading to blindness. Not caused by the HIV itself, but the inability of the patient to suppress a cytomegalovirus infection. So there are three views of AIDS now. One is by Bob Gallo, one of the discoverers of AIDS, and he says that if you're infected by HIV, that's all you need, get AIDS. Luc Montagnier, say maybe it's harmless except in the other cofactors. I think we're now veering towards the Gallo definition. But there are some naysayers still around. Duesberg and Mullis have been well known for saying that HIV really doesn't cause AIDS. And they've got, I think, a rather weak case. Kary Mullis got the Nobel Prize for PCR as a graduate of Dreher High School just down the road. He and Peter Duesberg, put forward the case that HIV is not the etiologic cause of AIDS. However, I don't think anyone other than Duesberg and Mullis really believe that any longer. The fact is that 50% of HIV-infected people progress to AIDS. No strong evidence at all that there is any other primary infectious agent that is causing this disease. If you have a patient who claims that something is the opposite, on our website there is a list of arguments to make that HIV does indeed cause the disease. So it's a retrovirus. We've sort of veered away now to patients and away from the etiological agent. It's a retrovirus. It's got a membrane. It comes from the host, just like all other retroviral membranes. It's got 3 genes, GAG, POL, and ENV. There's no oncogenic, no oncogene in HIV. 2 glycoproteins are encoded by the end gene. It's actually one primary translation product chopped up by Golgi body enzymes. The GP41 causes the fusion of the viral membrane with the plasma membrane of the cell. These are extremely highly glycosylated. These sugar chains act as an immune shield against the antigens in the protein part, and that's a problem in vaccine production. And then inside we have these group-specific antigens. We don't really need to know much about them. They just know they exist. The reason why I've just put up here in your notes the names of them, these are the things you see on the Western blot that's done to detect HIV in your patients. So there are these internal proteins that are coded by the GAG protein. And then there's the POL gene, and we all know what that does. Produces reverse transcriptase, integrase, and that protease that is vital to cutting up the primary translation product of the GAG and the POL genes into the mature proteins. Remember, one GAG primary protein, several internal antigens created during the maturation of the virus by this virally encoded protease, and that is the protease that the protease inhibitors act against. And they're very nice protease inhibitors because this is an aspartyl protease. It's not very like anything we have, and therefore these drugs can be selective. And we'll talk about that tomorrow. When we look at the genome of HIV, it seems such a complicated virus, such a complicated lifestyle. But it's no bigger than any other retrovirus. It's about 10,000 nucleotides. It does have some certain open reading frames in addition to what you normally expect. And they are indeed translated into proteins because we can find antibodies against these small control proteins here in the patients who are infected by HIV. So there's the GAG, the POL, and the end genes, just like any other retrovirus, but there are small open reading frames down at this end here that code for small proteins like TAT and NEF, etc. And you come across these when you're seeing articles about vaccines, etc., because some of these turn out to be very important, not in the structure of the virus, because they're not structural proteins for the most part. They're control proteins that this virus produces to cause its very weird way of replicating itself over a number of years. Yes, because that's going out through the endoplasmic reticulum and through the Golgi body, processing of proteins through that route are carried out by host cell-specified proteases, and the other two, the GAG and the POL, are cut up by the protease that's encoded here in the POL gene. Okay? So you actually have to cut the envelope glycoprotein before the virus buds out. The maturation of many of these proteins, the final cutting, actually takes place in the immature virus particle that's budding out of the cell. But it has to occur before the virus gets back to another cell or it's not infectious. So here it is. It's a picture actually I showed you yesterday. One of these rather boring little viruses budding out through the membrane here. But in vast amounts in certain cells. And all these virus particles coming out of this lymphocyte. So a retrovirus goes latent, specifically destroys cells. Why is it tropic for CD4 positive cells? Well, first of all, it fuses at the cell surface, so there's a cell surface receptor. It's rather like herpes virus. Doesn't need to go into an endosome. That results in syncytial formation. And syncytia may be of great significance in the spreading of HIV from one cell to another. It doesn't have to go out into the circulation and be endocytosed. Cells that are uninfected can fuse with cells that are infected. That's a consequence of syncytial formation. You get these multinucleate cells here. So that was important in vaccine production. Obviously, why is the CD4 cell targeted? Very early experiment done in the 1980s. HeLa cells are not infected. These HeLa cells, as you probably know, are a cell line derived from a person who had a cervical carcinoma. So these are cervical carcinoma cells, not infectable by HIV. Transfectin the CD4 antigen, which people had guessed at, was the unique characteristic that made these T4 cells infectable by HIV. You get infection. So that led to the idea that indeed the receptor was CD4 antigen. And that's why, particularly, you target T4 cells. But very early on, while people were doing this, there was this enigmatic observation that people really didn't know what the consequence would be of, what it really meant. And that was, you take a human cell here, a HeLa cell, put in the gene for CD4 antigen, the cells get infected. Take a mouse cell, put in the gene for CD4 antigen, they don't. So there's something that the cell's providing in addition to CD4. The mouse cell will bind the virus, but it won't get infected. So there's something in addition to CD4. It's more complicated than just virus coming in, having a GP120 virus protein on the cell surface that binds to CD4, and then endocytosis and infection occur. There's clearly a cofactor, a co-receptor in human cells that doesn't exist on mouse cells. Very important. This work was done in the laboratory of Bob Gallo again. What he observed is that CD8 cells produce a number of things called chemokines. I don't want to go into chemokines. You've probably heard more about chemokines than you ever want to hear again in the immunology lectures. But they produce these proteins called chemokines. And what Bob Gallo's people observed was, when you took these chemokines, they could block infection of macrophages by HIV, suggesting that the chemokine receptors, which are being blocked by excess chemokine, are also involved in the infection of the CD4 positive cell. And that, in fact, turned out to be the case. And we produce lots and lots of chemokine receptors on our immune cells. One of them is quite interesting, is CCR5. So the virus here will bind to a complex. Actually, it binds to CD4, but then to go any further, that CD4 has to associate with this chemokine receptor called CCR5. Chemokines, of course, will bind to this, and when you've got a mutant chemokine, which is rather interesting, even though you've got wild-type CD4, you don't get infected. So chemokine receptors are necessary cofactors along with CD4 antigen in the binding and internalization and infection by HIV of a CD4 positive cell. And this then takes us to the interesting people who don't get HIV infections. And there are some lucky people who don't. There are people who are long-term survivors. People who've been known around in the community who've clearly been exposed to HIV, but just don't get infected. Their CD4 cells remain normal. There's no evidence that the CD4 cell actually has been infected. Why? Well, this leads us to the chemokine receptor story, which we've just seen the beginning of. If you're fortunate enough to have CCR5 chemokine receptors that have certain deletions in them, and about 1 in 100 Caucasians, but no African Americans fall into this category, you don't get infected by AIDS. You can have unprotected sex with whoever you like, and luckily, you don't get infected by AIDS. So that shows how important these chemokine receptors are as co-receptors for with the CD4 antigen at bringing the virus into the cell and causing a productive infection. Now, people who are heterozygous mutants for this, and there are some African Americans who appear to have heterozygous mutants for this, they get AIDS more slowly. You have no idea why, because they should be able to, their wild-type receptors should be able to bind in this association with CD4, but it seems they do get infected, but it's a slower progression of the disease. So 25% of the long-term survivors are people who have deletions in chemokine receptors. There are other people who don't appear to have these deletions. We don't know why. But possibly other co-receptors are involved. There are a large number of these chemokine receptors. What I'm telling you is rather the surface story. It's clearly considerably more complex than this. Yes, it's probably the reason, indeed, why monkeys can carry these things without getting... Actually, monkeys get infected. Certain chimpanzees will get infected, but they don't go down with the immunocompromisation disease. The CD4 cells don't die off. So they seem to be able to... They probably fall into the category I'm going to talk about next, which are people who do get infected, but don't go down with disease. Okay? But chimpanzees, see, that's the problem with vaccine production. Chimpanzees can be infected with HIV, but you can't find any kind of manifestation of disease. Sorry, I interrupted you.
No. Do they still think the origin of this virus?
Well, there's a lot of speculation, but I don't think that speculation is well-founded as to where the virus came from. The virus came from a mutation in West Africa of SIV that was picked up by someone in West Africa as a result of close contact with chimpanzees. And if anybody tells you it came through polio vaccine, tell them it's a load of rubbish. Interestingly, this very same mutation in those chemokine receptors seems to be, it's called Delta 32, is one of the more common ones. Delta 32 was what probably protected people in the Middle Ages against the plague. Going back and looking at people who are descendants in villages in Europe, they can trace them back to the days of the plague. Clearly, these are the people, their ancestors are the people who survived. These mutations here, these deletions, are more common in those populations. It looks like this was protected us against another scourge several hundred years ago. A very interesting program on Nova, Nova about that a couple of years ago. Many chemokine receptors. This is a very complicated story. It's not as simple as I'm making out, but nevertheless, you can see what's going on here. And then there are the long-term non-progressors. These are people who have been infected with HIV, but keep their CD4 cells up. So these are probably a very heterogeneous group of people that are mounting a pretty good immune response. And there was this particular group here, a group of ladies in Nairobi whose profession was prostitution and still is prostitution. And they get pretty effectively exposed to HIV because 25% or so, at least, of their clients are HIV positive. And yet they don't seem to manifest the disease. They seem to have a very good immune response to the virus. They can clear the virus, and it's probably all to do with the sort of HLA antigens that they have in presenting the virus. HIV is a retrovirus. It has these various proteins that it carries with us, things like reverse transcriptase, the integrase, the protease, and of course, being a retrovirus, needs a primer. It carries a tRNA with it. It has 3 genes, no oncogene. But we've already seen that if you're a retrovirus, you do not need to have an oncogene in order to cause tumors. We saw about promoter insertion. This thing has got a very nice LTR. In the cells in which it's producing virus, it produces one hell of a lot of virus. So even though it doesn't have an oncogene, it still poses a problem as far as attenuated vaccines are concerned. Because we've seen putting that LTR in the wrong place can cause tumorigenesis. So if you want to have an attenuated vaccine, and work has been done on attenuated vaccines, then you've got to take care of this problem that a retroviral attenuated virus still has the potential to cause cancer. So it only grows in activated and replicates in activated T4 cells. Most of the infected T4 cells seem to die off, but they're replaced. Some revert to the memory state, and those are long-lived, and they don't replicate unless they become reactivated again. And you have this phenomenon also of clinical latency, which is just simply your patient gets infected and then 10 years later exhibits disease. So this can be called cellular latency, these properties here, and we have clinical latency, not seeing the disease for a decade or so. But remember, even though the patient may not be exhibiting any symptoms, the patient is still replicating virus and is infectious. So here's the idea then of what's going on in the patient here as far as this is simple immunology for people who are non-immunologists like me. An uninfected activated T4 cell. Dr. Mayer is probably absolutely appalled by this. He's thinking this is immunology for idiots. But anyway, an uninfected activated T4 cell just sitting around there, okay? As a part of the normal immune response, eventually most of those cells die by apoptosis. Some of them go into the memory T4 cell pool. These are very long-lived. Infected produces virus, because when they're activated, they produce virus. Most of those die, many of them because you're mounting a darn good immune response to the infected cell. Some of those don't get killed off, and just like any other activated cell, return to the memory state. And they are now chronically infected. They don't produce viral proteins, so they're not recognizable by the immune system because they're not activated. Only activated cells produce infectious virus. But the DNA's there in the nucleus. They're long-lived, 10 or more years. And it was this reactivation that then caused the disease to manifest itself again. So here you've got your memory T4 cell. You get an immune response, activation, virus production. And that caused a lot of gloom in the chemotherapy community, because at one stage we thought what you could do was treat the patient with the advanced heart combination of 3 drugs. The virus does not replicate under those circumstances. You can prove that in the patient. You're actually suppressing viral replication down to 0. And then all you have to do is wait around for the patient to clear out any infected cells, and these cells turn over, and then the patient will be clear of virus. But these cells, these memory cells, can last more than a decade. Some cells longer than that. And so for those cells to go away is going to take a very long time. And until those cells go away, you can't say the patient's is cured. Because if the patient goes off his drugs and some of these cells get activated again, out comes the virus again. And we see that. Unfortunately, some people ill-advisedly stop taking their heart medication, and back comes the virus. It's probably coming back from these memory T4 cells. But we know that in the presence of heart, there is no replication of virus because we don't see the signature mutation of the virus that normally occurs. And mutation, as we've seen with the retrovirus, is an inevitable consequence of having the way in which it replicates its genome. So why do all these T4 cells disappear over the 10 years or so the patient has left if it's not treated? Well, there are some reasons why. First of all, those cells that are infected just burst. They get lysed by the virus. They're just millions and millions and millions of viral particles per infected cell get produced, and the cell just dies. So that's one reason. But most of the cells aren't actually infected. So that's not the whole answer to why the CD4 cells go away. You can have that syncytium formation that I mentioned before, where you've got an infected CD4 positive cell, which is also GP120 positive for the viral antigen. Along comes an uninfected cell. The CD4 of this cell binds to the GP120 of this cell. And because they can fuse at ambient pH, you get syncytial formation. That doesn't really explain it either. You can get cell-mediated immune response. That'll kill off infected cells. But most of the cells aren't infected. It's a sloppy virus. It has a short half-life. Bits of GP1 of the virus fall off. The GP120, when it is cleaved, when the GP160 is cleaved to GP120 and GP41, the 2 component parts, it's the GP41 with the fusogen that is the transmembrane protein, and the rest of the surface protein, the GP120, is just held on by non-covalent associations with GP41, and it falls off. So one of the hallmarks of an infected patient is a large amount of free GP120 circulating in the bloodstream. This GP120 comes along and binds to CD4 antigen, and then the cell can be recognized as looks like an infected cell and can get destroyed. But probably the reason why the CD4 cells go so much is because of the body inducing apoptosis. Now, we don't know a lot about this in CD4 cells, but we're learning something about it. We know more about it in CD8 cells. What happens in CD8 cells when they go away is this. The virus can bind, and other things can clearly bind to the chemokine receptors, and in the case of CD4 cells, also to the CD4 antigen. And when you've got a CD8 cell here, what you get when the virus binds is the production of a thing called TNF-alpha receptor. And I'm not going to ask you questions about this. This is just for interest's sake. In the macrophage, you get the binding of TNF-alpha. So here in the macrophage, you've got the formation of TNF-alpha. Here's the receptor. They get together. The macrophage sends a death signal to the CD8 cell, which commits suicide. So it looks like the macrophages, when they're stimulated by HIV, can actually induce the other cells of the immune system, the CD8 cells and the CD4 cells, to give up the ghost and commit suicide. And that may be the explanation as to why so many of the CD4 cells go away, and in the later stages, the CD8 cells go away. Now, these macrophages, as I said, are the way in which you acquire HIV if you get it by sex. They can pick up HIV just like the T4 cells because they have CD4 antigens. They're CD4 positive cells, but they can get the virus in another way, via the Fc receptor. Because if the virus is bound antibodies, such as anti-GP120, it can be picked up by the Fc receptor. And that can be shown experimentally. Putting the Fc receptor into cells, they can also get infected by HIV. That's a problem for vaccines. Predisposing the virus to opsonization and infection of macrophages. All these little things about the biology of HIV make it an enormous challenge to the vaccine industry to produce an effective vaccine. And so far, it's all been quite a failure. There's no good vaccine. Many have been tested, and all have been found wanting. So the macrophages are this sort of Trojan horse that brings in the virus into the body. The first viruses that come in are macrophage tropic, because macrophages also have a chemokine receptor. They actually have the CCR5 receptor. It infects, you get macrophages in the semen if you're transmitting it via that route. The macrophages get infected, and they also produce, as a secondary effect, changes in your cytokine levels. And that might be the basis of some of these slim disease things that you see in particularly African populations, where the patient also loses weight very, very strongly. And slim disease is very much like Visner, which was the archetypal lentivirus, and that also infects macrophages as its primary cell host. They also form a reservoir outside the blood. They're also the cells that can take HIV into the brain, and then brain cells get infected. And the mature macrophages can just go on budding out virus for a long period of time. So we look at HIV, we look at AIDS, and we say CD4 cells, and of course CD4 cells are the reason. The loss of CD4 cells is the reason why the patient goes down with immunocompromisation disease. But the macrophages and other cells are also infected, and they have an importance. Now, I stressed this last time, this business of population polymorphism. And amongst retroviruses, we've already seen, this results from two things. One, it's using a host cell enzyme, like all other retroviruses, to duplicate its genome. Normally, that enzyme makes messenger RNA. Messenger RNA is used a couple of times and degraded. If you've got a mutation in one of your messenger RNAs, it doesn't matter. If there's a bit of misreading when POL2 transcribes that gene, it doesn't matter if there's an occasional amino acid that's wrong. That messenger RNA is not going anywhere. It's not going to be perpetuated. It's going to get degraded. But here, it is being perpetuated. It's the enzyme that perpetuates the genome of the organism, and so mutations accumulate. Not only do you have a problem with POL2, this host enzyme, reverse transcriptase doesn't proofread either. So there are two reasons here, because of the way this retrovirus duplicates its genome, that you get a huge mutation load in the virus. Now, in most cases, that mutation load is going to be detrimental to the virus, and the virus will just die. But some of them give the virus a selective advantage, particularly in the hostile environment of the bloodstream where antibodies are being produced. Pol2 has an error rate of about 1 in 10,000. 1 in 10,000 of the nucleotides it puts into the messenger RNA, or in this case into the genome of HIV, is the wrong nucleotide. Genome of HIV is about 10,000 nucleotides long. That means that every time you replicate the DNA, it's different. To replicate the RNA, rather, it's different. Every time. Every single virus will pick up a mutation. Every possible mutation in that virus with this huge production of viral particles occurs in your patients every day. An enormous population of variant viruses as a result of being a retrovirus. And a lot of double mutations arise as well during the day. And that means that the virus that you see later on in the patient is very different from the virus that infected the patient in a number of ways. And that's a problem for developing vaccines. Most vaccines haven't even been raised against macrophage tropic HIV. It's been CD4 tropic HIV. And we know it's coming in through the macrophages. And most vaccines that we've studied so far have been subunit vaccines, taking GP120, GP41, those sort of proteins, putting them into the patient, saying, let's get an immune response here, that'll kill off the virus. But the virus is constantly mutating. And as we saw, the immediate reaction of the host is not the humoral response. The reason why you kill off the virus is a CTL-mediated response. And we know virus can spread from cell to cell without ever coming out into the circulation. So a good vaccine is going to be a cell-mediated response vaccine. And it's going to have to be a vaccine that will be active against this hugely variant population. So here, the initial infecting virus is macrophage tropic. It actually has the CCR5 as its co-receptor. These turn out to be non-syncytium-inducing agents. Most vaccines have been made against the T4 cell tropic strains. As it mutates, there are an enormous number of subtypes here. If you acquire the virus rectally or vaginally here, you're getting a macrophage tropic one, bound by dendritic cells, taken to the lymph nodes. The tropism is constantly changing as the co-receptor affinities are changing because of this mutation. As a result, you then change the co-receptor that acts with CD4 in the T4 lymphocytes, and so you've got this other one here, very common, it's CXCR4. A virus that is changing daily. It's a pretty hard target against which to design a vaccine. So I've more or less said this. Early infection, macrophage tropic, non-syncytium, slowly replicating late infection, T4 tropic, syncytium-inducing and high virus titers. The most variable protein is GP120. An enormous number of mutations arise. There are parts of GP120 that don't seem to be able to bear a high level of mutation. They are essential parts of the virus of the GP120 that if mutated, become non-functional. The CD4 antigen binding site is a good example of that. So these are regions of the GP120 against which perhaps we could design some kind of immune attack. Because as the virus mutates, the ones that mutate their CD4 binding site tend to just disappear because they're not highly infectious. They can't bind onto CD4 antigen anymore. So maybe we can, these conserved regions of GP120, but other parts of and if it turns out to be the really exposed parts of GP120 are constantly mutating. And if you look at a picture, it may be in the notes, I'm not sure whether I put it in this year, there are these variable regions, there are these constant regions. The constant regions, the ones that don't seem to be able to bear this mutation rate, They're very highly glycosylated. They're protected against antibodies by sugar chains. Unfortunately, we're now seeing another phenomenon that is a problem for vaccines. That's hybrid HIV. People who've been infected by more than one strain of HIV to form mosaics of HIV. And that's caused, of course, another increase in the variability. There are actually two strains of HIV. There's HIV-1 and HIV-2. HIV-1 is the cause of the major pandemics throughout the world. HIV-2 also causes the disease, but it rather more slowly causes the disease, and it's largely limited to West Africa. Not only, though, does the GP120 acquire all these mutations that allows it to escape the immune response, all of the proteins are getting mutated. The transcriptase gets mutated, and that allows the virus to escape the effects of drugs, which we'll talk about tomorrow, that attack the transcriptase. We've got some pretty good drugs that inhibit reverse transcriptase now. Very good drugs. The problem is they're very effective at the beginning, and then mutation arises as the virus that escapes the drug duplicates itself, and you get resistant mutants. And that's the whole problem. Retrovirus is antiretroviral drugs, a long story of resistant mutants. In fact, that's also all virus drugs have this problem of the development of resistant mutants, but particularly in retroviruses, because they've got a higher mutation rate. So you get the drug problem as a result of that. Also, the protease is varying as a result of this polymorphism, and that gives us inhibition of the resistance arising against the protease inhibitors. The only way to stop population polymorphism developing is to stop duplication of the virus. If we can stop duplication of the virus, there's no population polymorphism. And that's what the heart regimen does. It actually reduces the amount of virus particles per day down to undetectable levels. Therefore, provided you get it down to undetectable levels and you stop the virus reproducing itself, then you have a very good drug regimen. Singly, most of these drugs are not very useful because they don't suppress replication 100%, and therefore the surviving virus mutates and then forms a resistant mutant. So we know now what the basis of this high polymorphism. We get various subpopulations. Now, there are other cells. We talk about CD4 cells, we talk about macrophages. There are other cells in the body that get infected by HIV. We won't go into these very much, but endothelial cells in the brain, certain other brain cells. One of the consequences of an HIV infection is age-related dementia. Epithelial cells in the bowel and vagina also get infected. These, for the most part, are CD4 negative cells. And the reason why they get infected is there is another receptor for HIV, and it's a lipid. It's a galactocerebroside. So if you've got on these cells a particular galactocerebroside, then they can become infected as well. So that's probably the route by which these cells become infected. So what are we to do? 20 years of HIV infections, 20 years of AIDS. What are we to do about it? Clearly, we have no vaccine, and it's probably going to be a decade or more before we have a decent vaccine, despite the huge amounts of money that's been poured into vaccine development. There have been bright spots in vaccine development, and they've turned out to be failures. There was one that was a very bright spot a couple of years ago, based on our knowledge of the Nairobi prostitutes, because these are people who fight off the infection. But when a vaccine was developed, it was not effective at all, based on that. There are some people who have mutations in some of these control proteins. Maybe they could be the basis of vaccine development. A vaccine was developed involving one of these control proteins. Turned out that in many people, In fact, it wasn't given to people, it was given to chimpanzees that made the problem worse. And vaccinology is replete with examples where vaccines actually make the problem worse. So vaccines are no good. So what we've got at the moment is education. Fortunately, HIV, despite its reputation, is not very infectious. You've got to take steps to get HIV. If it were as infectious as influenza, and we could get it by somebody coughing in our face, we would be in dire trouble. But it's not highly infectious. Sexually transmitted, so therefore obviously education programs have been one of the great boons in reducing the spread of HIV. Chemotherapy, as I've said, has been very useful. You get resistance, suppress replication, no resistance because of no replication. We've got the highly active antiretroviral therapies. These consist usually of 2 nucleoside analog reverse transcriptase inhibitors and 1 protease inhibitor, or 2 nucleoside analog reverse transcriptase and 1 non-nucleoside reverse transcriptase inhibitor. And we'll look a bit more at those tomorrow morning. Does HIV cause AIDS? I've already alluded to the answer to this question. There's only one single common factor between a gay San Franciscan in the 1980s, a New York drug abuser, an African heterosexual, a hemophiliac, AIDS, resulting in these populations, HIV infection. The one common factor. That is pretty good evidence that HIV causes AIDS. The fact that the spouses of hemophiliacs also get AIDS when they become infected by HIV. Children of hemophiliacs and drug users get AIDS when they become infected by HIV. HIV has preceded AIDS in every population in which AIDS has arisen around the world. Only HIV. Some people argue that Koch's postulates, you've probably come across Koch's postulates, have not been fulfilled. It's one of the arguments made by the camp that says HIV does not. Well, Koch's postulates, one of the Koch's postulates, you take the organism, you inject it into, you isolate the organism, you inject it into an animal or patient who has not received the organism, and you look to see if disease results. If we have purified disease, inject it into a patient, does that patient get down with AIDS? That's pretty good proof that HIV causes AIDS. Not many volunteers came forward to do this. However, we do have that Koch's postulate proven now. We know that simian immunodeficiency virus, which probably gave rise to the HIV in human populations from chimpanzees. The chimpanzee SIV is very, very similar to the strains of HIV found in West Africa. Injection of purified simian immunodeficiency virus into monkeys gives you simian AIDS. As a result of laboratory accidents involving the mishandling of purified HIV, HIV has been injected into humans, and those humans have gone down with AIDS. So, it looks like pure HIV can indeed cause the disease. Remember though, education has leveled off the infection rate, HIV slowed the death rate. That means the number of infected people is rising, and education is our best bet at the moment to decrease the amount of spreading of the disease. The drugs are very expensive. They've been very successful in Western countries. As I was telling one of the people over here, the average annual expenditure of some sub-Saharan countries on healthcare total is less than $25 per head of population per year. Until a few years ago, heart cost $12,000 per year. It's gone down enormously now, down to a few hundred, and many people, but a small minority, are getting the heart therapy. For the people who don't, which is the majority of the population in sub-Saharan Africa, education is the only way to go. Okay, I'll see you tomorrow morning. We'll talk more about drugs that are active against viruses.